not only is it a foggy Friday, but today will be a fishy Friday. So I'm going to tell you about sort of zebra fishing for genes. And a couple general topics that we'll cover is exactly what are zebrafish and why they are studied. Some things that we have already learned from them, and a little bit about what the future might hold for using zebrafish and the future of scientific research. So up here we've got a couple zebrafish, and you're welcome to come look at them a little bit after the talk. But I also have a little movie, so you can see they're striped. That's why they're called zebrafish. Their genus and species name is Daniorerio, but most scientists still just call them zebrafish. And they are at the Exploratorium, so you might have seen this actual tank if you visited the top floor there. So they are a freshwater tropical fish, which means that they live in warm waters without salt. They're usually found naturally in rivers in India, but they are very prevalent in fish stores around the US, so you also might have seen them in that context. So we study zebrafish for several reasons, and there's always advantages to using a particular organism or system in research. And some of the advantages, step by step, are, for one, they're vertebrates. So I have a cladogram or an evolutionary history showing our closest ancestor, chimpanzees, were pretty similar to them. And if you look at the vertebrate tree, fish are on the vertebrate tree. Okay, so we have a lot of characteristics that we share with fish, and much more that we share with fish than, say, plants or other organisms that are also in the world of biology and research. So they are close to humans. This word fecundity refers to the ability of these organisms to reproduce in massive amounts. So about 100 to 200 embryos are produced per clutch. And a clutch is essentially the progeny or offspring from crossing. And that's sort of what we call setting up a mating between a male and a female zebrafish. And this tank that I have up here is a crossing tank, so they don't live in here all the time. You just put them in there when you want a male and a female to mate or cross. And this kind of shows what happens. The female lays her eggs, and the male puts his sperm on it, so they're externally fertilized in the water, and they get collected down here, and you can collect them and watch them develop. The kind of neat thing about this is that it happens on a light cycle, so as long as they're in a period of dark and a period of light, they're pretty much set up to mate just like they would in nature, except we can manipulate it in a laboratory. So once they're laid, they have a very short generation time. So that means from essentially the time that they're fertilized to the time that they can also go on and produce their own offspring is only about 3 months. So it's pretty fast. And they also develop rapidly. So they actually have most of the organs that they will have in about 48 hours or 2 days. So from the time that they are fertilized, 2 days later, they have lots of organs, and I'll show you an example of that. They're easy to maintain, so we have them here at City College. You just got to pretty much make sure that they have water and food, and that light cycle keeps going, and they'll cooperate most of the time. And then last, which is sort of one of the most neat things about zebrafish, is that they have these translucent embryos. So here is a 48-hour-old zebrafish embryo, and they kind of look like a fish thing, maybe sort of alienish, but they have, again, many organs already. So the eye is very obvious. The heart, I'll show you in a couple slides later, but it's pretty much right here underneath the eye. This big mass of sort of yellowish orange stuff is its yolk, and just like chick embryos have yolk, or if you eat eggs, there's yolk in there. It's the energy source for these growing zebrafish. So they have to get their nutrients from somewhere because they're not connected to any other organism, and they can't quite eat until they have their mouth formed and all their digestive system formed. So this is where they get their food until they can get it on their own. Along their back are these kind of chevron-shaped or triangle-shaped structures called somites. And somites are what end up becoming our back muscles and our vertebra. and you have the spinal cord inside of it, right? So a couple of things. There are the other organs that you can't see just by looking at a fish under a microscope, but they have a liver, a pancreas, sort of stomach equivalent, and things that work like kidneys as well. So many organs are ready by 48 hours. This is the heart that I told you I'd show you. And so you can see it beating. And that's, again, one of the really great advantages to using these translucent embryos. So just putting a zebrafish under a microscope, you can watch how the heart beats. And it has only one atrium and one ventricle. But again, if you understand what happens on an atrium and ventricle, you can also relate that to our heart, which has 2 atria and 2 ventricles. Older, so day 3 or 4, this is an image of the actual blood cells moving around in the trunk. So again, because they're transparent, you can actually see sort of, it's a bit blurry and just should link. Yeah. But every single stream here is blood circulating. And you can see there's sort of big vessels going down and back, as well as little ones going out to the side. So they're transparent, and you can see the heart and the blood vessels. And another way that they're used is to use another organism's genes and place them into zebrafish. And so in jellyfish, they naturally produce these fluorescent molecules. And so here are just 2 pictures of sort of the range of colors that you can get in nature from jellyfish. So you've got reds and yellows and blues. This is a bit bluish greenish. And using those genes, you can create transgenic organisms. So this is called fluorescent marking, or using fluorescent markers. And that's really, really exciting when you start making essentially fish that glow in certain areas, and then watching them under a microscope and creating all these movies that I'll show you more as we go. So green fluorescent protein and red fluorescent protein are the ones that you'll see most today. There are other colors, just like you saw in the zebrafish picture, so blue and yellow and purple, but green and red are the most commonly used. And again, you can make transgenic zebrafish. So that's taking the genetic information from jellyfish and inserting it into the genome of zebrafish. So the way that it can be really taken advantage of is to put it into a tissue-specific place. So for example, the heart or just the blood vessels, and that way you can watch just the heart under a fluorescent microscope, and you drive the expression of GFP or RFP in that particular tissue. And then, again, if you take a microscope, and some of the more technical microscopes are confocal microscopes, so they just have a bit more detail, and they're designed to look at this sort of fluorescent imaging. And you end up with 4-dimensional images. So not only 3D, as in length, width, and height, but also over time. And so that's sort of the really useful tool that zebrafish provide. You saw the blood vessels before, and it was just kind of a blur of cells moving through. But here we have the blood vessels in green, and then the actual blood flowing through in red. So again, watching sort of how this forms, and also I'll tell you later when things go wrong, we can learn about development and how vertebrates form different tissues. So developmental biology is where I learned how to use zebrafish. So most of my examples come from that field, and then at the end I'll tell you about other uses for using these great fish. But to give you a little bit of an idea about what exactly is developmental biology, it's sort of twofold. So one part is differentiation, and every one of us is different from each other, and likewise every piece of our body is different from another piece. So our heart has unique characteristics, and our toes have unique characteristics. And at some point, you start out with one cell, and it has to become all these different parts. So forming many different types of cells and tissues from the one-cell zygote, that's differentiation. And the other thing that happens during development is morphogenesis. So morphology is sort of the shape of an organism or anything, really. And it's as you go from one cell to the fish that you see up here, things have to change shape and also organization. So you've got some things on top, some things on the bottom, and they might even flip their orientation. Cells have to divide, and you have to get more of them. So growth is involved with changing shape. And cells can move. So you might have a cell on one side of the embryo, and it has to move all the way to the other side. So all these things happen, and the goal of developmental biology is to understand how it happens and how it's regulated. So here is a zebrafish that is at the 2-cell stage. So someone set up a male and a female at night. In the morning when the lights turned on, they crossed and laid a zygote. And so the yolk is again here. So it starts out with lots of fuel. And then up here are 2 cells. So they didn't quite catch it at the 1-cell stage. It happens pretty fast, so you got to be quick under the microscope. But 2 cells is quite good. And as you watch, you'll see what I talked about. So division. No, this is about 15 hours total from start to finish. Okay, so cells are moving now, right? They're going down, and they involute or rearrange their position. And now you can kind of see sort of the body plan. So you got eyes up here. These are the somites, and it starts to form the tail. We'll watch it one more time for awe effect. Yeah.
You're going to talk about how you insert the porous protein genes in specific tissues in a minute?
I don't, so I can answer. So essentially, you take a 1-cell stage or close to 1-cell stage.
Pluck a cell out.
Well, no, you like the first slide. You start with something that looks like the first slide, and you can you would have to inject a piece of DNA into the yolk, and
into the yolk.
Yeah, and so there's sort of a high level of failure, essentially, because that piece of DNA has to insert itself into the genome. And there's ways of tricking the genome and the DNA to make it more efficient, but usually it takes about 500 injections to get a stable insertion.
Tissue-specific?
You would have to have it driven by a tissue-specific promoter. So, so whatever is upstream of the gene, right, turns it on and off. And so if you have that sequence and you know, for example, the heart, it's a heart-specific myosin, and the promoter is going to drive only expression of GFP in the heart. So you put your fluorescent protein after the promoter. So I might have said a lot of lingo there. Does that make sense? Or?
Well, I understand what you're saying. When do you do that?
So you'd have to get a back. So the genome's been broken up into small pieces and put into bacterial chromosomes. So there's like a library. And the zebrafish genome is mostly sequenced. So if you know you have, like, many genes are known nowadays, right? So you know you have a heart-specific gene, and you search for that gene among the bacterial chromosomes. The bacterial chromosome is like 100 kilobases long, and so most likely it includes some of the promoter. So you take that bacterial chromosome back, and you flip out your gene for GFP, and that way you have the promoter and your GFP, and then you have to inject that whole back into the zebrafish at the 1 to 2 cell stage. So it's I didn't talk about that because it's a lot of technical stuff. So for most of us, we can just say it's magic and it happens, but those are the technical aspects of it.
Yeah.
Okay.
Okay, thanks.
Sure. Yeah. Well, I mean, you get 200 embryos per clutch, right? So if you inject 3 days in a row, hopefully you get one. Yeah. Okay, so fundamentally, no matter what you're talking about as far as function in any organism goes, you start from DNA, and then you make the RNA, and that RNA gets translated into protein. And whatever the protein is going to do gives you the function, right? So in the case of developmental biology, we're just looking for genes that regulate the function of differentiation or genes that regulate the function of morphogenesis, right? So those are sort of the two categories that make up all these processes that happen from zygote to organism. And it all starts with DNA. And that's when we get into the title of my talk, Zebrafishing for Genes. So making transgenic organisms is sort of a tool that helps you do the zebrafishing for genes. And the way that you might have heard about making mutants is using what's called reverse genetics. So you can go forward or you can go backwards. And the reverse thing is, again, what most of you might have heard of with knockouts or mouse knockouts or whatnot. So if you know a gene, okay, so again, lots of genomes have been sequenced, and there's a lot known at this point about different genes, and you want to figure out what happens if an organism doesn't have it, then you remove the genetic material. And again, that's called a knockout, and I don't go into the technical details of that either, but it's kind of similar to what I just mentioned. And then you look at what happens. So with a little more graphics that might help you. So you've got lots of DNA, lots of chromosomes, and one of them is your gene of interest. And so let's say you're interested in your heart development, so you know something in the heart, and you take that gene and you get rid of it, and then you watch what happens to that organism. So the genetic information is not there. The flip of that is what's done with zebrafish, okay? So again, reverse genetics is making a mouse knockout or taking the gene out. But forward genetics is sort of, you have a pool of mutants, and you make those mutants randomly. So I'm going to go forward just for a moment. So you've got, again, a lot of DNA and a lot of random genes that we may not know what they do, and we want to figure it out. And so you expose the random genes to a mutagen that can change the sequence a little bit. Okay, so now it says quandam gene, so it's changed somehow. It wasn't the same as it was before. And then you're going to go and let them develop and look at them. So you end up with a characteristic or a phenotype of interest. And again, it's not just one, as it is in this picture. It's hundreds of thousands of genes that might be affected, and you end up with a big pool of mutants, and you want to figure out which ones are interesting to you, and then you work backwards and figure out the gene. Does anyone have any questions about that? So essentially, you're fishing for genes. So you make a lot of random mutations, and you look for things that are interesting, and then you have your gene, and you try to figure it out. You have a question? Yeah. Oh, it's just changing the letter R to Q. So I was just trying to say that the sequence is changed a little bit. So it's just a made-up change. So I could have written rantum gene or random peen. So just a little change instead of removing the whole thing. Okay, so we fish around for genes, and again, this is not something that you can just do overnight. So these are called forward genetic screens, and they take months to years to kind of iron out all the possible mutations that can come out. And hopefully one of them is interesting enough that you spend a lot more time focusing on just one instead of the hundreds that come out of the screen. So some of the things that might come out is, again, here's a wild type or a normal zebrafish, and then after it's been mutated, it looks a little bit different sometimes, right? So this one is the same age, yet it's much shorter, and this little bubble here is representative of a heart defect. The same bubble is over here where the heart is on this zebrafish. If you look at the size of the eyes compared to the size of the eyes there, it's much smaller. So mutants can come in many different shapes and sizes. And again, you get lots of different ones. And so if you were interested in heart development, you might want to spend some time looking at this mutant or this mutant and investigate it further. But you get a lot of options, and you try to select the one that has the characteristic of interest to you. Those were visible just with the naked eye, but if you use the transgenic zebrafish, this is actually a picture of a liver, a pancreas, and a stomach of a zebrafish. So using transgenic zebrafish, you can look at that under a microscope as well. You don't have to pull off the yolk and figure it out. You can just look. And these are two examples. So this one is just flipped, as in its right side is its left side. And this one has a morphological defect. So the liver spreads across both sides, or the pancreas spreads across both sides, and it looks much different. So you get a mutant that you find interesting. So again, if you were interested in heart, you could have chosen one of the heart ones. If you were interested in the liver or the pancreas, you would choose one of those. And then you work and figure out what gene is mutated and spend a lot of time trying to figure out what exactly that the protein does. And some things that have been accomplished or learned from this process are actually quite extensive, but a couple examples is the vertebrate plan across the left-right axis. So if you drew a line down my body, right, I have pretty much symmetric structures, but my heart is much more skewed to one side, my liver is skewed to one side, likewise your stomach is not distributed exactly equally. So you have a bit of sort of morphological difference between right and left. So here might help you to see the liver and the heart and lungs are also different. So I mentioned heart development. You've got different heart mutants. I'm just going to kind of brush over those, but a lot of children, human children, are born with heart defects, so it's a definite area of interest. And then angiogenesis is new blood vessel growth, and that's an area of great interest for cancer research. So you have a tumor. The only way that tumor is going to survive is if it's getting blood and oxygen and nutrients from blood vessels. So if you can deprive it of its nutrients, you can hopefully prevent it from growing. And it also applies to other blood vessel diseases. So macular degeneration is essentially the back of your eye, your retina is gradually dying over time, and it's one of the leading causes of blindness. So I showed you some vessels before. Maybe you can let me know if you can't see it. But here are the bigger sort of arteries and veins, and then the little vessels. And what you'll see is sort of a sprouting of a vessel, and that represents new ones growing. So again, studying the process might aid us in treating cancer. So you can see the little threads coming out, forming new vessels. And then one that I worked on a lot is related to the neural tube. Okay, so the neural tube is eventually what becomes your brain and your spinal cord. And all vertebrates have a brain and spinal cord. And you have neural tube defects, again, just like heart defects. They're of concern because they affect a large percentage of human births. So abnormal development of the neural tube, some examples are spina bifida and anencephaly. So these can lead to paralysis or possible death. And again, they affect a substantial amount of births annually, so about 2,500 births annually. And you might have heard about this nutrient that can prevent neural tube defects, folic acid. And it's definitely pushed in the media, and there is a lot of research to support it, but no one really understands what it does or why it works and prevents neural tube defects. And I don't have any answers to that, but I just thought I'd mention that to hopefully bring some pieces of your life together. The mutant that I worked on did have a neural tube defect, and just like there's a process to going from one cell to an adult zebrafish, there's a specific process in forming the neural tube called neurulation. And it's, again, specific to vertebrates. And you essentially start with a flat plate, so just a sheet, and it folds up on itself to form a tube. And I'll show you a picture on the next slide. So you've got the formation or the starting formation of a tube up here. So those sheets are folding up and starting to form a seal on the top and leaving sort of a tube or a hole in the middle. And from a cross-sectional view, you can see the same sort of thing. So this started out flat, and then the edges rolled up, and eventually will form a circle and a tube. So the neurulation in zebrafish happens pretty similarly. So up here is a flat sheet. You can kind of see it's outlined in yellow, and it comes to the middle. It doesn't fold up quite as obvious, but it is quite similar in process, and you end up with, again, a tube with a hole in the middle. So neurulation, something can go wrong along the way, and that would lead to a neural tube defect. This is the back or the dorsal view of a zebrafish. So, if the fish was, or an embryo, if the fish was facing into the wall, these are the cells, and you can see they do kind of round up and reach towards the center as the fish grows. So it's almost like it's folding down. So this would be the center and where the tube would eventually form. Okay, so you end up with something that kind of looks like the top in 3D. And a lot of the images I'll show you are either sliced sagittally through, so you can just see the line in the center, or sliced transverse, so that you see the circle with the sort of slit lumen. And if you think about a cat's pupil, it sort of has the same shape. So it's not a round circle, but a slit lumen. That lumen forms what are similar to our brain ventricles. So we've got these fluid-filled spaces. And this is a sagittal view, and you can see that the fluid starts to fill in. So it's expanding as the fluid pumps in and fills that ventricle space. And this is a cool picture. No? It's my favorite, actually. But this is, so you can see, this is the head of a zebrafish with that tube forming along it back. And just in case you're like, what does this have to do with humans anyway? There's a photographer, Leonard Nilsson, that does some really amazing images using special microscopes and lenses. And so here's a zebrafish head with its neural tube. And here is a human embryo with the same structure. So it's kind of shocking how similar it looks. And likewise, from the side, you have brain ventricles in zebrafish, and they're sort of outlined in red here. And here's a human embryo with the same sort of space that exists on their brain. So the mutant that I worked with looked like this as far as its global features go. So you might notice that it also had a heart defect, but I focused on the neural tube component. And for comparison, there's always a wild type or a normal embryo and then a mutant one. So if you look at the shape back here, it almost looks like two triangles full of fluid in those ventricles. And the mutant has a uninflated or sort of flat ventricle, so it doesn't fill with the fluid. And likewise, tried to show it in a little more obvious. You've got the fluid-filled space in white, and then it's lacking in the mutant. So cross-section-wise or transverse section-wise, what I found was that that lumen, or the slit in wild type, is continuous. So it's like a line, and it would go all the way down the embryo. But in the mutant, it sort of had pieces, so it never connected. It's most probably obvious in this picture. So there's a little piece on top, in the middle, and on the bottom, and they don't connect completely. So the formation of that slit or that ventricle area was not occurring properly in the mutant. This might also give you an idea. So that slit, sometimes it's together, sometimes it's incomplete. So in wild type, it would be a straight line all the way from the first picture to the last picture that you saw. So the gene, so I had this mutant, and it was interesting, and ultimately we wanted to know what gene was doing it. So I'm not going to get into the details of this because it's also quite technical, but you do some positional cloning and genetic mapping, and I found that this was the name of the gene, so partitioning defective 6 gamma B. but the great thing about it is that the abbreviation is party 6 gamma B. So my poor lab had to listen to all these party jokes whenever I gave any presentation, and I'm like, let's get the party started, and the party is, yeah, so I have tortured you as well. But it plays a role in apical basal polarity, so it is kind of known what it did already, but no one had ever observed this neural tube phenomenon in a vertebrate, so it was studied in Drosophila and other systems, but so it forms the lumen or the hole or that ventricle, and also orients cell division. So again, that hole, right, didn't form properly, so it makes sense. A couple more movies about cell division. So this is a forming neural tube, and maybe I can turn the lights down to see a little better. So there's a couple cells that divide, and it's on a loop, so you'll see the same thing over and over again. But right here, you can watch the cells round up in the middle and then divide across the center. And again, this movie was taken using fluorescent molecules, right, with a living embryo under the confocal microscope. So you can't do that with really any other vertebrate system. So this division, where it crosses the midline, sets up where the ventricle is going to form. So it's kind of at an angle because it's alive and I couldn't get it perfect. But as it divides, you're going to end up with something right in the middle. And this might be a better image of just one. So you can focus on just that one, right? It rounds up and divides across the side. So what I found was in this mutant, since the neural tube ventricle or the ventricle space didn't form properly, the divisions were not in the right orientation. So essentially, I'll just show you this movie, and there's a cell division that happens right here. So instead of going across, it's at an angle. And also affected the morphology of the neural tube. So from the mutant that I worked on, starting again with that forward genetic screen, finding something interesting about it, we learned a little more about vertebrate neurulation. So it was a novel phenotype that no one had ever seen before. There a little more about how the morphology of brain ventricles form and how the orientation of cell divisions can shape organs. So, you know, if cells divide in the wrong orientation, you can end up with a whole bunch of cells in a space where they shouldn't be. So the orientation of how a cell divides can play a role. And hopefully it'll be a future tool for studying neurulation in vertebrates. I have a little more, but are there any questions?
This defect, is it equivalent to spina bifida?
It's not equivalent to spina bifida, no. So spina bifida is when the tube doesn't close on the top, and most of those tubes are closed on the top. Yeah?
My background's in art and design, so I'm kind of like someone who understands counterfeiting. You know, you study the original to recognize what's off.
So you always want to compare to wild type. So that's a standard across any science is you always want to have something as a control for comparison. So there's these mutations are recessive. So whenever you mate the fish, only a quarter of the offspring will be mutant and 75% will be wild type. So you always have sort of an internal control built in, and you always want to include mutants in any movie or assay that you do in the case of fish. And then as far as what mutant you choose to work on, that can be sort of, if you want the scientific answer, you want something that is interesting to what your lab works on and will improve the knowledge of science. So you want something, that you can contribute and hopefully will, again, improve the knowledge of developmental biology. But as far as actually being a person, you want something that's going to get you data and get you results. As being a grad student, you want something that's going to help you finish within a timeline that's reasonable. So yeah, you're going to choose something obvious because you know you're going to get something out of it. One hazard of choosing something obvious is I also worked on another mutant that was involved with splicing. No one cares about splicing. Well, some people care about splicing, but it's not as sexy as biology people like to say. So that's sort of the cool topic of the year or month. And splicing just isn't interesting to a lot of people. So I didn't tell you about the splicing mutant because it wouldn't be as nice of a story. So there's a bit of science and a bit of more non-objective choice in making that decision. Yeah. Yeah.
So when you're crossing, you always have a single pair of fish. Is that right?
Well,
if you get a really messed up mutant, you'd never be able to get.
Yeah. So part of the process of the screen is cleaning up the background so that you end up with a fish that only carries this one mutation. And you can have a family, so a family of heterozygous carriers. So you don't end up with just 2 fish. If you only have 2 fish, you're kind of risky, right? Because if one of them dies, you can't do anything. So you want to have many. And the wild type component of it will be a variable in your control all the time, but that's also more realistic to living organisms in general. Does that answer your question? Okay, good. Okay, so just a little bit about the future, briefly, what we might learn from zebrafish in the future. So other ways that they're used that are becoming more and more popular are pharmaceutical screening. So biotech companies and making drugs is a very lucrative option in science. And they are, again, you get a lot of fish from every mating. So you can put them in little plates singly and expose them to possible new drugs or new targets and observe if it affects their heart or blood vessels or any organ of interest. Effects of chemicals in the environment. So there was a recent study about atrazine, which is an endocrine disruptor that's used as a very popular crop. Is it an herbicide or an herbicide? In agriculture. So the zebrafish were exposed to atrazine, and it was proven that it affected their endocrine development. So hopefully more information will come out if needed. And then we have embryonic stem cell research. So, heart transplants, and I'll tell you a little bit about beta cell transplants. So you have an embryonic stem cell right here. We've seen it several times. It is embryonic. It's a stem cell. It's going to become everything that an adult fish has. So doing this sort of research, in a way, is doing embryonic stem cell research. It's not human embryonic stem cell research, but it's definitely similar. And again, a zebrafish has a pancreas. So you've got liver, gallbladder, pancreas, and I've tried to focus in on this part here and here. That's the part that makes insulin. That's the beta cell component. So in case you don't know, type 1 diabetes is related to insulin production, which comes from your beta cells, or in the case of fish, that little circle. And people that have type 1 diabetes can't make enough or negligible amounts of insulin. So they have to treat themselves by using injections pretty much multiple times a day or wearing an insulin pump. And so there's a lot of precedence that beta cell transplants could be used to treat and cure or eliminate the need for insulin injections. So it's been done with cadaver transplants and shown to be effective for several years. So there's, again, a lot of precedence for it. But the only problem is we need to be able to make these beta cells that hopefully your body won't reject. And so again, you've got a stem cell that ends up forming a pancreas. So what we learn through studying zebrafish can hopefully be applied to human embryonic stem cells and how we end up with a pancreas and possibly those beta cells. Any questions about that? No? And then a last sort of note is to glow or not to glow. So this was maybe 5 years ago. These particular types of transgenic zebrafish were in the news quite a bit. So they are transgenic and they express the fluorescent protein everywhere. Okay, so it's not just in the heart, it's everywhere. And when you expose them to UV light, they glow. So it's kind of a cool pet to have. But the transgenic organisms bring up other sort of ethical issues that we might be able to learn from. So it's, again, a gene that's inserted into another organism. And I showed you how useful they are for research. So all this live imaging and these images that we took from perhaps not alive embryos sometimes too, and marking cells, it's a great tool. But at the same time, it's also very controversial for the environment. So we don't know what happens exactly when we put these transgenic organisms out in the environment. In the case of zebrafish, they probably won't do anything. They'll probably not even survive here locally. If you think about putting them in the bay, is really cold. They're not going to live. It's salt water. But the state of California is a no-glow state. Okay, so glowfish cannot be purchased or sold in the state of California. And in a way, it's
That's not research, though, right?
No. What?
Even for research?
Oh, you can have them in research, and you have to put these signs up that say, like, do not remove from facility. So yeah, you can have them at research facilities. But it set a precedent because, again, zebrafish are probably not going to harm things locally. But if these were not outlawed in the state of California, where do you draw the line? And that's part of becoming scientifically educated and thinking about things, is learning where to draw the line. And so California drew the line here. Other states allow these fish to be sold, but sort of a neat quote that I saw to kind of end with pretty much is that science tells us what we can do and gives us a lot of information, but we have to decide what it is that we should do. So hopefully I've at least told you that zebrafish are an excellent tool for studying developmental biology, and there are always people to thank. So this was my advisor in grad school and a lot of people that helped me do the work, and of course the City College Biology Department for inviting me and welcoming me here. So I'm happy to answer any other questions that you have.
Frankenfish.
Okay. Thanks.